In this lecture, we're going to talk about insulin. We're going to talk about oral hypoglycemic agents, and we're going to talk about glucagon. So if you think about it, insulin is secreted from the beta cells of the pancreas. The alpha cells secrete glucagon, and insulin is a drug given for the treatment of diabetes. And oral hypoglycemic agents are agents which reduce the blood sugar. And glucagon is a counter-regulatory hormone of insulin. Meaning if insulin reduces blood glucose, then glucagon increases blood glucose. So that's why we call it a counter-regulatory hormone. So it makes sense that these three be studied together. And that is precisely what our lecture today aims to do. So the objectives for this lecture is we're going to look at introduction. Specifically in introduction, we're going to look at how the body handles, how the human body handles glucose. So basically the physiology of glucose absorption. And then we're going to look at the biosynthesis of insulin. Okay, subsequently we're going to talk about diabetes. We're going to see what is diabetes. We're going to look at what are the different types, what are the symptoms, and what are the possible complications if we don't treat diabetes. And that would set the stage for us to learn about the treatment goals for a diabetic patient. Then we would be classifying insulins. We would be briefly looking at their pharmacology. And we would do the same thing with oral hypoglycemic agents. Subsequently, hopefully, we would look at self-monitoring of blood glucose, and we'll study the pharmacology of glucagon. And hopefully, at the end of all this, we will end up with some pearls which we can use in our practice. Let us look at the first question. How does the body handle sugar? How does the body handle sugar? So to answer this question, we need to ask a question. How does sugar enter the body? Okay. Basically, there are three routes by which sugar enters the body. So when we say enters the body, we mean enters blood. Okay. So now we are going to look at entry of sugar into blood. Okay, as we mentioned, there are three ways by which sugars enter into blood. What are the three ways? The first way is through the diet. The second way is by Okay, what is glycogenolysis? Right, it is the breakdown of glycogen. What is glycogen? It is a polymer of glucose. Which is biosynthesized and stored in the liver. Okay? So the muscles as well as the liver produces glycogen, but it is the hepatic store that is used to bring glucose into the blood. Okay, not the glycogen produced by the muscles. Okay, so it's a polymer of glucose which is predominantly stored in the liver. Okay, so let's add a note here and say that muscle glycogen is not used for releasing glucose into blood. Okay? Okay. The number 3 source is Gluconeogenesis. Okay, so what is gluconeogenesis then? It is using non-carbohydrates as a source to generate carbohydrates. Okay, so in integrated metabolism, we say that the end products of digestion of fats, proteins can be converted to glucose and vice versa. Okay, so gluconeogenesis is also the source of how blood sugar increases. Okay, so let's take the first one and try to understand more about it. Okay, so that's number 1, that's number 2, and that's number 3. Okay, so there are 3 sources of how sugar enters into the blood, and we are now going to look at diet. Diet. So what do we mean by diet? Diet is basically the food that we eat. Okay, and the food. So in the food, what is the source? The most common source is sucrose and lactose and starches. Okay? So sucrose and lactose, if we think about it, they are disaccharides. Okay, and what are starches? Starches are basically polysaccharides. Okay, disaccharides and polysaccharides. What is common to both disaccharides and polysaccharides is that both these are made up of monomeric units. Okay, both disaccharides and polysaccharides are made up of monomeric units. And what are these monomeric units called? They are called, if this is disaccharide, that's polysaccharide, the monomeric unit obviously would be, you're absolutely right, monosaccharides. Okay. Okay. So these monosaccharides, we're going to ask the question, how do monosaccharides become disaccharides and polysaccharides? So monosaccharides bind to each other by losing, okay, they lose by losing proton and a hydroxyl group. Okay. And this is called, this reaction, what is this called? This is called a condensation reaction. Okay. Why is it called a condensation reaction? Because two monosaccharides, they are going to lose water, so their entire structure becomes reduced, and therefore we call it a condensation reaction. Okay, so in a condensation reaction, what is there? There is loss of water. In the body, this condensation reaction is reversed. Okay, so this is reversed in the body. Okay, so addition of water is called hydrolysis by the reaction called hydrolysis. Okay, and in the, because it's happening in the body, you need water as well as you need enzymes. Okay, so it's an enzyme catalyzed hydrolysis. So what type of hydrolysis? Enzyme catalyzed. Okay, and you need water obviously. Okay, and that happens in the body. Okay, all right. So are these the only sources of carbohydrates? No. There are also other carbohydrates. What could be the examples? Right. Amylose. There is glycogen. Alcohol. Lactic acid. Pyruvic acid. Dextrins and pectins. Okay. So these are the other carbohydrates that are again used as a food source. But most commonly, the source is sucrose, lactose, and starches. Okay. And starches, depending on where your geography is, previously, before travel became common, we could say that what would be the major source of starch in geographically isolated populations. For example, in say China, it would be predominantly rice. And if you look at Africa, maybe it would be predominantly cassava, which is a root. Okay? And if you look in the West, it would be predominantly wheat. Right? But then this is fluid. Okay? Now our food habits have changed. We travel around the world, and so that's not strictly true. But once upon a time, we could know. Right? Okay, and now let's look at this Now that we are with starches, let's look at starches. Okay. So starches, the way they are digested in the body, the digestion begins in the mouth. And which is the enzyme in the mouth? You're absolutely right. Ptyalin. Okay, that's the enzyme that, almost digests 20% of the starches. And subsequently, it goes into the stomach, and after it reaches the stomach, it reaches the intestine, where we have the pancreatic amylase. Okay, and this basically converts the starches into maltose. Maltose and around 2 to 4 glucose polymers. Okay, polymers containing of about 2 to 6 glucose units. Okay. This maltose is acted upon by maltase and alpha dextrinase, okay, which are kind of enzymes present in the small intestine. And this maltose is converted to glucose. Okay, those polymers also get converted to glucose. Okay. Now, we have talked about lactose and sucrose. Let's see what happens to lactose. Let's take another color. All right, so lactose, which is a common polymer that we consume as a part of our diet. It's a milk sugar, lactose. So this lactose gets converted to glucose and, yes, galactose. Okay, so that is why lactose is called a disaccharide. It gets hydrolyzed to galactose and glucose. And the enzyme, yes, lactase. Okay. So what about our other polymer that we were looking at? Do you remember which polymer? Yes, sucrose. Okay, again it's a disaccharide, meaning it would be hydrolyzed to another subunit, and that is glucose and fructose. Okay, so what is lactose here? Let's just write it down before we lose track of it. So lactose is basically milk sugar. Okay, sucrose is what? Cane sugar. Fructose, it's present in fruits and yeah. Okay, but what's important is why are they very commonly present in our diet? Because milk is consumed, as well as cane sugar, sugar beet, okay, the sweet potatoes, which are sugar beet in fact, they are the source of sucrose, and therefore lactose and sucrose is the most common source of glucose. Okay. Now these enzymes here are sucrose with which enzyme? Sucrase. Okay. So these enzymes, we have sucrase, we have lactase, we have maltase or alpha dextrinase. Okay. These enzymes, let's write it down here again. Maltase, alpha dextrinase, lactase, and sucrase. Okay. These enzymes are collectively called alpha glucosidases. Okay, why is that important to know? We'll come back in just a minute. Okay. So up till now, what are we talking? We are talking about how sugars enter the blood. Okay. How does sugar enter the blood? That's what we have considered. Okay. So now let us see, how do sugars exit the blood? Okay. How do sugars exit the blood? Okay, let's zoom a bit. Okay, how does sugar exit the blood? Okay, so that is what now we are going to talk. Okay, so sugars have now, we have consumed carbohydrates, digestion has happened, and the sugar has now entered our blood. So we are basically concerned about glucose when we say sugars. Okay, so because we are writing all these examples, so that is why I did not specifically write it as glucose. Okay, but ultimately what's important is all these things get converted to glucose, and glucose is the main sugar. Okay, so now we are looking at how does glucose exit the blood. Okay, so you might already know, how does it exit? Yes, some of it is stored. Okay, what is the storage form of glucose? Right, storing glucose takes up lots of space. So therefore the body has come up with a clever idea of using a polymer instead of glucose itself, and the polymer is glycogen. Okay. It is stored as glycogen. Okay. How does it exit? What is the other pathway by which it exits the blood? Right, there will be utilization. Okay. How is it utilized? What is the main metabolic pathway by which glucose is utilized in the blood? Very good. Glycolysis. Okay. The other thing that happens is, we said that the primary metabolites are interchangeable. Okay, so they can be converted. So there can be conversion into other forms, like, which is the storage form of energy? You're right, they can be stored as lipids. Okay, what else can happen if there is lots of concentration in the blood? Okay, and it has reached the renal threshold. And what is the renal threshold? Greater than 180 milligram per deciliter. Then it will be excreted through the urine. Okay? Okay. So why is all this important for us as pharmacologists, okay, or as practitioners? Okay, so the idea is, if our goal, okay, so we have said that there is blood, there is entry, and there is exit. Okay, there is entry of sugar into blood, and there is exit of sugar from the blood. Now, if suppose there is an increase in blood sugar and we want to decrease it, okay, so this understanding gives us an idea that either we can reduce entry or we can increase exit. Okay, so our idea is, if suppose blood sugar, here we mean glucose, increases in the blood. Okay. So what is our option? What can we do? What can we do about it? We can reduce the entry, or we can increase the exit. Okay. And because now we know the physiology of how it enters and how it exits, we will have targets. Okay. So let us illustrate that with an example. Okay. So here we said that starches are absorbed as glucose by the action of alpha glucosidases. So our aim is to decrease the entry, decrease absorption. So what can we do? Can we block alpha glucosidases? Definitely yes. So if we block alpha glucosidases, what's going to happen? Complex starches will not be converted to glucose. Okay. And if there is less absorption, definitely less sugar will enter the blood. If less sugar enters the blood, definitely blood sugar level will go down. Okay. What about the exit? Okay. So if we give something that increases the storage of glycogen, then the blood sugar level will come down. Okay. So how can we promote storage of glycogen? By giving insulin. Okay. Similarly, we have different targets. Okay. So that is the importance of knowing the physiology of how the body handles blood sugar. Yeah. All right. So blood sugar level is controlled within a tight range. Okay. Blood sugar level is controlled and maintained within a range. Okay, what is that range? We'll be discussing later on in our lecture. Okay, what is the numerical range? Okay, but for now we understand that blood sugar level is controlled and maintained within a range. And this maintenance is called glucose homeostasis. So the first question is, so how does it maintain homeostasis? So if there is increased blood glucose, body secretes insulin. Okay. If there is decreased blood glucose, the body secretes the counter-regulatory hormone glucagon. Okay, so this is a brief, this is a very general overview of glucose homeostasis. Okay, so the point to remember here is why does the body control and maintain glucose within a range? Okay, so that is the important question here. So it is controlled and maintained within a range. Why? Okay. So there can be two conditions: increased blood glucose and reduced blood glucose. So increased blood glucose, what is it called? You're right. Hyperglycemia. And in the same vein, what is reduced blood sugar called? It is called hypoglycemia. Okay? So hyperglycemia can interfere with the vasculature. What are vasculature? The blood vessels. Okay, and the heart. Vasculature and the heart. So together, this is called the cardiovascular system. The brain needs glucose for its metabolic activity. Okay? It needs glucose. It cannot survive without glucose. So hypoglycemia is detrimental to the brain. Now, if you think about it, if your heart is compromised and if your brain is compromised, your survival cannot be guaranteed. Okay? So, there can be, the liability can be, and this can compromise the viability of the human organism. Okay. So these both conditions can become a threat to life. Okay. So that is why the body tries to actively control and maintain blood sugar within a very close range. Okay, because to preserve the preserve life. Okay. Right, which means that there can be increased morbidity. Okay, what do we mean by compromise the viability? It means there can be increased morbidity and increased mortality. Okay, so that is why the body tries to maintain the glucose level within a very narrow range. Okay. Now, we want to know whether there is a problem. Okay, how do we know? How can we, if there is a problem? Okay, if there is a problem. Okay, and we want to check. Whether the, what do we want to check? Whether, okay, we want to check whether the body is able to, maintain homeostasis. Okay. So if we want to do this, we need to check if the body can decrease blood sugar when lots of sugar is entering the blood. Okay, when is lots of sugar entering the blood? It is the fed state. Okay. Then we need to check whether the body can increase blood sugar when blood sugar is below normal. Okay. So what is that state when blood sugar is below normal? That is called fasting state. Okay, so we define fasting as 8 hours without calorie intake. Okay, there is no restriction on water intake. Okay, so to look at whether there is a problem with the maintenance of homeostasis of blood sugar, we check the status in the fed state as well as the fasting state. Okay, so that is how we diagnose whether the person is having diabetes or not. Okay. So this introduction, along with the discussion that we are going to have next on the biosynthesis of insulin, will set the stage for our discussion on clinical aspects. Okay? Let us now look at the biosynthesis of insulin. Okay. So, insulin is a peptide hormone. It's a peptide hormone which is synthesized in the beta cells of the pancreas. Okay. So let's orient ourselves as to where the pancreas is located. Okay. So let me very diagrammatically try to draw what we are talking about. Okay, so let's orient ourselves. So this is the stomach, and this is our depiction of the pancreas. So this is a leaf-like organ that sits somewhere in like this in relation to the stomach. So this pancreas has got two roles. It's got an exocrine function and it has got an endocrine function. Okay, so exocrine meaning the secretions which are released through a duct, and this duct is what we are showing here. It opens in the this C-shaped region of the duodenum. So the first part of the small intestine is called the duodenum. So into the C part of the duodenum is where the hepatopancreatic duct opens, and that is the exocrine part. Now, if we take a section of the pancreas and we blow it up, we blow up this section, and we are trying to draw what we would see in a histological section. Okay? So what we would see is this rounded kind of cells which has got a lumen, and these cells kind of have got their walls opening into the lumen. So these are all basically depiction of how a typical cell would look like. And these cells obviously would have the nucleus. Okay, so those are the nuclei that I'm trying to draw here. Okay, so cells, these cells, this singular is called acinus. Okay, so we have drawn acini. Okay, acini is the plural of acinus. So what is acinus? These are cells which are typically present in a pancreas. Okay, and the acinus forms the exocrine part of the pancreas. Now there is another sort of area that is seen within the pancreas, and within this different sort of an area, we see a different type of cells. Okay? So these different types of cells, the different type of cells that we see, they are called alpha cells. Okay, the other one would be beta cells, and we have delta cells, and we have cells that are called F cells. They are also called PP cells. Okay? So these cells, the beta cell, as we have mentioned earlier, it secretes what we are interested in today, insulin. Okay? The alpha cells secrete glucagon. The delta cells secrete somatostatin. And the PP or the F cells secrete the pancreatic polypeptide. Okay. So now if we look at this diagram, we would notice that this area kind of looks like an island. Okay. Island in the Greek is called insula. Okay. So it's from insula that the word insulin has been derived. Okay, so that's just a small trivia. Okay, so this insula contains the endocrine part of the pancreas. Okay, so this insula makes up the endocrine part of the pancreas. And there are 4 cells that are present in the insula, which are beta cells, alpha cells, delta cells, and F cells or PP cells. Okay, so there you go. That is, so the beta cells secrete insulin. Okay? So let's look. We have told that insulin is a peptide hormone. Let's look some details of how this peptide looks like. Okay, so this peptide is made up of 2 chains. Okay, there is a shorter chain which is called the A chain, and there is a longer chain which is called the B chain. Okay, what is this chain? This is a chain of amino acids. Okay, so inside we've got amino acids which are linked to each other by the peptide bond. Okay, so this is a block representation of what essentially is a chain of amino acids. So the A chain is made up of 21 amino acids, AA meaning amino acids, and the B chain has 30 amino acids. Okay? So the A chain and the B chain are linked to each other by disulfide bonds. Okay, so there are two disulfide bonds. So two disulfide bonds link A chain with B chain. So two disulfide bonds, this is the first one and this is the second one. And what is observed is there is also a disulfide bond within amino acids present in the A chain. So this is an intra A chain disulfide bond. Okay. So if you look at the specifics, it is between the 7th amino acid, let's write it here, B7, 7th amino acid. So this is 7 meaning amino acid 7. Okay, amino acid 7 of B chain and 7th amino acid of the A chain, and that's the first disulfide bond. The other one is B19, okay, the 19th amino acid of B and the 20th amino acid of A chain. Okay, they form, so it's not, it's kind of staggered, and if this is 7 and 19 should somewhere be around here, but for making the diagram a bit more, visually appealing, I have put it near to each other, but you get the idea. Okay, so the 19th amino acid and the 20th amino acid is linked by a disulfide bond, and which the bond which is supposedly somewhere here, we have represented it here, but we have not sacrificed the accuracy of it. Okay, so it's the 19th amino acid that is linked to the 20th amino acid, and this is seen between the A chain, the 6th amino acid of A chain, and the 11th amino acid of the A chain. Okay, so they form a disulfide bond, but then that is within the A chain. Okay, it's not linked to the B chain. Okay, so this is what insulin is. It is a 51 amino acid peptide hormone. Yeah. All right. It has a molecular weight of, I think, 5808 Dalton. Okay. So the molecular mass is about 5808 Daltons. Okay. So this immediately gives us certain information. Okay. So as far as drugs are concerned, we classify drugs as small molecules and large molecules. Okay, so small molecules meaning approximately about 650 Dalton to 1000 Dalton. Okay, so and larger molecules obviously is greater than 1000 Dalton if we use 1000 Dalton as the cutoff. So if we look at this information along with that, we understand that insulin is a large molecule. Okay, so insulin is a large molecule drug. Okay, now, and it is a peptide. Okay. If this information is taken together, then we understand that we can infer that the route of administration of insulin would be parenteral. Parenteral, one, because the molecular weight is so, and secondly, because it's a peptide. Now, if we give it orally, the peptide would be degraded by the proteases. Okay, so it cannot be given per oral. Okay, because it can be degraded by proteases, which are most common in the small intestine. Okay, so this is the reason why the route of administration would be parenteral. Okay, so the most commonly administered route of insulin is subcutaneous. It is administered subcutaneous. Okay, yeah, and there is a soluble form of insulin that can be administered IV. Okay, but for all the others, it is the subcutaneous route. We will visit this this information again. Okay, so just hold on to this thought. Okay, so now we are going to look at how we end up with insulin. Okay, so we're looking at the biosynthesis within the cell. Okay, biosynthesis. Okay, so biosynthesis is basically how the body makes insulin. So that's why we call it biosynthesis, the body being bio, okay, being living. All right, so insulin, so the cell synthesizes insulin. Where does the cell synthesize insulin? What is the information? The information obviously is the DNA. And the DNA, in this case of insulin, is contained within the 11th chromosome. Okay, so let me draw my depiction of chromosome. And this is chromosome 11. And the chromosomes, we know it has got two arms. One is the short arm, which we call the P arm, and the Q arm. Okay. So the information encoding insulin is a gene that is located in the short arm or the P arm of chromosome 11, and that gene we call it because it is insulin. We take the first three words, and genes are usually represented by capital letters. So we put INS and we say that gene codes for insulin. Okay. So this gene, where is it located? It is located in the short arm of chromosome 11. And the locus would be, if you write the locus is 11P, okay, and it is 15.5. Okay, so that is the locus of the INS gene, which codes for insulin. Okay, all right. So now let's look at the gene products of INS gene. So INS gene, it is expressed. What do we mean by expressed? It means when the DNA is converted to protein. Okay, that process we call expression. So when this gene is expressed, what we obtain is something called preproinsulin. Preproinsulin. So let's diagrammatically represent preproinsulin. Okay. Let's call this preproinsulin. Now, a preproinsulin would have two ends. Okay, it would have a carboxyl end, let's say, and N end. Okay, because amino acids have got amino group and a carboxyl group, so it should have a carboxyl end and a amino end. And let's call this the amino end here in this diagram. Now, there is a sequence of amino acids at the N end, which, let's say, we can color this up. Okay. N end, there is a sequence of, let's write it here, sequence of 24 amino acids. Okay, so this length is basically 24 amino acids, and this is what is called the signal peptide. So the first biosynthetic event that happens is the preproinsulin. From the preproinsulin, there is removal of the signal peptide. So let me write down here, preproinsulin minus signal peptide. Okay, so from the preproinsulin, this signal peptide is removed, and what do we get here? We get what is called the proinsulin. Okay, so preproinsulin minus signal peptide, we end up with proinsulin. Okay, so let's draw the other parts of this sequence. We will have the B. Okay, so this is the, it's depicted by another color. Okay, so we have got this part. Okay, and this part is the B chain. Okay, so the B chain is how many amino acids? 30 amino acids make up the B chain. Okay. Okay, so this part is the B chain. 30 amino acids. At the end is where we have the A chain. Okay. So let's color it again. Okay, and let's label it. This is, if that is a B chain, this is the A chain. Again, B chain should be longer and A chain should be shorter. So let's modify this. Okay, so hopefully we have made it shorter, and that's the carboxyl end. and this we call the A chain. Okay, again, how many amino acids? 21 amino acids. So, when INS gene is expressed, it is expressed as a long chain containing 4 parts. Okay, what are the 4 parts? We have part 1, which is the signal peptide. Part 2 is the B chain. And part 3 is what connects the B chain to the A chain. So we call this the connecting peptide. Okay, so it's also called this C-peptide. And this is made up of 31 amino acids. Okay, so if this is 30, next is 31 amino acids form the connecting peptide. And the A chain is again 21 amino acids. Okay, so what is proinsulin then? Proinsulin would contain only these 3 parts. Okay? Okay, so I think we can draw this again. We can copy this. Or let's draw it fresh. Okay. Okay, so we are trying to diagrammatically show that the pre-proinsulin has become proinsulin. Okay, so this is pre-proinsulin. Okay, and this is proinsulin. Okay, so this is proinsulin, and that is preproinsulin. Okay, so this preproinsulin, what happens to preproinsulin? So let's draw this how we see it in the body. Okay, so this proinsulin, we are going to draw a folded depiction. Okay, the same thing, but it's kind of folded. So it's a diagrammatic representation. And what we see lined up is, this is the B chain. Okay, so first we drew the B chain, then we drew the A chain. So when we fold it, this is the B chain. Okay, that is the B chain, and that is the A chain. Okay, so this is a folded diagram. Okay, depiction of folded proinsulin. So now if we fill in the details, we can find that there is a disulfide bond between these two. Okay. And there is a disulfide bond between the amino acids in the A chain. So this part is the A chain. This is the A chain, and that is the B chain. Okay, so that is the A chain and that's the B chain. So then intuitively we know what is going to happen. This proinsulin, it will lose the connecting peptide. Okay, so proinsulin, when it loses the connecting peptide, it becomes insulin. So proinsulin minus connecting peptide. Okay, what do we end up with? We end up with insulin. Okay. So this is the sequence of events that take place within the pancreatic cell. Now let's put more information here. Okay. So what happens is, first proinsulin, let's draw up a cell, and this cell is going to represent a pancreatic cell, and we're going to tell this story again. So we have got the nucleus. Let the nucleus be somewhere here. Okay, and let's represent that as chromosome 11. Okay, it expresses what? Preproinsulin, and within this cytoplasm, this preproinsulin will be converted to proinsulin. Okay, this proinsulin will be packaged into a vesicle. This proinsulin will be packaged into a vesicle. What will happen to proinsulin within the vesicle is the proinsulin will lose the connecting peptide. Okay, so what we would see is insulin molecules. Okay, so this is a dipeptide. We'll see an insulin molecule and we will see connecting peptide within the vesicle. But this connecting peptide is now separated from the insulin itself. Okay. Right. So this proinsulin continues to become insulin, and with the removal of C-peptide. Now, initially what happens? Two insulin molecules which have lost the C-peptide will form a dimer. Okay, it will form a dimer. It will form a dimer, means this two will aggregate together to form a dimer. Subsequently, what happens in the vesicle is the pH of the vesicle. So the pH of the vesicle decreases. Okay, pH of the vesicle decreases. Then what happens is there is secretion again of zinc molecules. Is then 2 plus. Okay. There is first event is there is reduction in pH. The second event is secretion of zinc ions into the vesicle. Okay. So these are events happening inside the vesicle. So there are the other event that happened was conversion of proinsulin to insulin plus C-peptide. Okay, subsequent to this, what happens? These two happen. Okay. After this, step 1 happens, then step 2 happens. So there is secretion of zinc ions into the vesicle. Now what will these zinc ions do? Okay, so now we're going to look at the role of zinc ion. So this zinc ion, okay, 2 zinc ions, let's say this is 2 zinc ions, this will combine with 6 insulin units. It will combine with 6 insulin molecules. Okay, so all this blue representations, so this blue things here represent insulin molecules. Okay, so 6 units. So how many units? 6 units. Okay, let's number them. 1, 2, 3, 4, 5, 6. So 6 units of insulin combines with 2 zinc molecules, and this is called a hexamer. So the insulin forms a hexamer. Why hex? Hex means 6. Okay. Hex means 6. So now there are 6 units. Okay. All right. So that form in the vesicle. Okay, we are talking about an hexamer forming inside the vesicle. This hexamer crystallizes within the vesicle. Okay, so inside the storage form of insulin is a crystal. Okay, insulin, zinc, crystal. Okay, it's crystalline. So what is the advantage of crystallization? So why does it crystallize within the body? It crystallizes because a crystal is most stable. Okay, so stability is high. Okay. The other reason is it is compact, meaning very less space is required for 6 units of insulin to be found. So it's very compact, meaning there is less space required. Okay, what is the advantage of that? More and more insulin molecules can be stored within a single vesicle. The other reason is that a crystal, basically it is most stable, so it also prevents proteases from degrading the peptide. So within the body, within the cytoplasm, there are also proteases. Now these proteases can also sometimes enter into the vesicle, and if it enters and it encounters a free insulin molecule, it will immediately metabolize it to amino acids. Okay. And once insulin loses its structure, it won't have activity. Okay, so the point is, insulin requires this structure. Okay, if the disulfide linked double chain structure is required for its activity. If insulin by some chance loses the overall structure, then it won't be active. So if a protease comes, the protease will come and will degrade all these amino acids. Then insulin, the action of insulin won't be seen. So it's very important that the newly synthesized insulin be protected from proteases which are within the cell. Okay. So that is why within the pancreatic cell, insulin is present as crystals along with zinc molecules. Okay. Okay. Then what happens? Now we are going to talk about a pancreatic cell. Okay, so this is a pancreatic cell. What happens? How does it release insulin is now what we are going to discuss. Now, this is a pancreatic cell. Okay, pancreatic cell. Now, this pancreatic cell has got certain channels. Okay, so these are channels, protein channels, specific for glucose. Okay, so it has got a special name. These channels have got a special name, and that is, it's called a Glute transporter. Okay, so glucose transporter, that's how Glute comes. Okay, so glucose transporter. Okay, so Glute is a type of transporter protein, and there are different types of Glute. So in the pancreas, Glute 2 is present. Okay, so what's the function of Glute 2? It enables the entry of glucose. Okay, so we are now talking about a normal pancreas. So what's the function of the normal pancreas? When blood sugar increases, it should release insulin. That is the function of a pancreatic cell, a normally functioning pancreatic cell. Now, in the blood, glucose level has increased, and that increased glucose level brings the glucose in proximity with GLUT2 transporter, and the GLUT2 transports the glucose within the pancreatic cell. Now what will happen is, this glucose in the pancreatic cell will be metabolized, meaning it will generate ATP. Okay? Now there are certain other channels on the pancreatic cell, and one of the most important channels which is present is potassium channel. Okay, so this potassium channel transports potassium from within this cell to outside, because the intracellular ion is potassium. Okay. So if potassium goes out, it will basically hyperpolarize the cell. Okay, so we don't want that happening. Okay. So what happens is, this ATP goes and blocks the potassium channel. Okay, blocks the potassium channel, which will depolarize the cell. It depolarizes the pancreatic cell, and this depolarization event opens up another channel. That channel is calcium. So calcium ions from outside will now enter the cell. These calcium ions will make this vesicle to move downwards. Okay, where this vesicle will fuse with the membrane of the pancreatic cell. It will fuse with the membrane of the pancreatic cell, and there will be exocytosis. Okay, exocytosis of what? There will be exocytosis of whatever was present within the vesicle. What was present? Insulin hexamer. Okay, C-peptide. Okay, and then there was the pH was less. So all the contents of this vesicle will come out. Okay. This insulin hexamer which comes out immediately reaches where the pH is higher than that of within the vesicle. Inside we said the pH was low. Now it is encountering a higher pH. Okay, higher pH. Okay. And it is also encountering bicarbonate ions. Okay, bicarbonate ions. Okay, so where is this pancreatic cell releasing it? It is releasing it into blood vessels, into blood. So in the blood, there is the pH of blood is higher than that of vesicle, and there is also bicarbonate ion. So the increased pH and bicarbonate ions will convert the hexamer. In the presence of pH and bicarbonate, the hexamer is converted to monomer. Okay, so now there are insulin monomers in the blood. Insulin monomers. So these are insulin monomers. The biological activity is due to insulin monomers. Okay? And these monomers then go and bring about their action in other cells. Okay? So overall, this is the regulation of insulin release. So whenever blood glucose increases, the pancreatic cell releases insulin, and that insulin monomer acts on different cells, bringing about the action of insulin. Okay, so let us trace this story after that. So these insulin monomers come into the blood. From the blood, they reach different tissues. The most important tissues are liver and the adipose tissue and muscles. Okay. Muscle tissue, let's write it, muscle. Let us look at these tissues one by one. So for that, let's draw a liver cell. Okay, so this is a representation of a liver cell, and that's the nucleus. Let's label this hepatocyte. So hepatocytes have got again this transporters similar to pancreas. It's called Glute 2. Okay. Glute 2. Okay, similar to, if you remember, similar to pancreas out here. Okay. So the important thing to remember about Glute 2 is Glute 2 is insulin independent. Okay, it is insulin independent, meaning it doesn't require insulin to work. So in pancreas too, we saw that contains Glute 2. Okay, so this is insulin independent. So the trigger for Glute 2 or the activation of Glute 2 is entirely depending on the glucose which is present in the extracellular fluid. Okay, similarly for hepatocytes. Okay, so that said, glucose enters into the hepatocyte. So hepatocytes, they also need to grow, they need to maintain their integrity, they need to maintain their enzymes, so they require energy. So a bit of this glucose is converted to ATP that does all that. The rest of it goes and becomes glycogen. Okay? So glycogenesis takes place. So all in all, in the liver, what happens is, because there is generation of ATP, we can say that there is increased activation of glucokinase. Okay, so if you remember the glycolytic cycle, for the generation of ATP, you see that glucokinase happens to be the most important enzyme. And then there is glycogenesis, so there is increased glycogen synthetase. Okay, and then what we observe is that there is also inhibition of gluconeogenesis. Okay? So inhibition of gluconeogenesis. Okay, so what is gluconeogenesis? It is the production of glucose from non-carbohydrate sources, which may be lipids, which may be proteins, so on and so forth. Okay, so it decreases gluconeogenesis. And what happens to the lipids? So there are also lipids within the hepatocytes. So what it does is it increases lipogenesis. Okay, so there is no breakdown of lipids. Because if there is breakdown of lipids, then we find ketone bodies. So there is no ketones. Okay, so there is no ketogenesis. So basically there is inhibition of ketogenesis. So we can write here inhibition of ketogenesis. Okay, so what is the problem with ketones? Ketones can depress the CNS. Okay, so it is life-threatening. Okay, so there is inhibition of ketogenesis. Okay, then the other thing, which is the other metabolite. So the other metabolite would be proteins. Okay, so there is decreased proteolysis. Okay, so metabolism. So what is metabolism then? So we have insulin. It has got metabolic effects. Okay, so by metabolic effects, we mean the interchange of biochemical components, but to simplify, we say all the biochemical processes involved in converting food to ATP, okay, and all the pathways that are involved in this process usually are what we term as the metabolic effects. Okay, so those are metabolic processes. So insulin has got metabolic effects, and here we're talking about effects on the primary constituents of food, which are carbohydrates, proteins, and fats. Okay, so we said carbohydrates, all these things are involved. Okay, so this is about carbohydrates. Okay. Okay. And lipids, what happens? There is increased lipogenesis. Okay, so that fat portion of it. Okay, so what happens to fats? And what's the other? The proteins. So what happens to proteins? Okay. Right. So what we observe again here is that there is increased lipogenesis. So there is production of lipids. Okay. So there is production of lipids and decreased proteolysis, but it stimulates production of proteins. Okay, so if proteins and lipids increase, that will lead to weight gain. Okay, so increased production of proteins and lipids, it is an anabolic process, meaning there is a buildup. Okay, so we can say here that insulin has got also anabolic effects. Okay, and it is the anabolic effects, as we have seen here, that leads to weight gain. Which also means if there is no insulin, then the opposite of all these processes is going to happen. There is decreased glucokinase, there is decreased glycogen synthesis, there is increased gluconeogenesis, and there is decreased lipogenesis, and there will be formation of ketone bodies, and there will be increased proteolysis. So people with insulin deficiency would become emaciated. Okay, they would become thin. They wouldn't show a normal weight. All right. Okay, so we have talked about the metabolic effects and anabolic effects in the hepatocyte. Let's take up the other target tissue of insulin. Okay, and here we're going to talk about adipose tissue. Okay, so let's draw an adipose tissue here. Say adipose tissue. So we said again, adipose tissue has got another type of transporter. Okay? So mind you, here it is Glute 2, whereas in adipose tissue it is Glute 4. Okay? And also in muscle tissues. Okay, so let's do our muscle tissue as well. Okay, because it's corrected here. It is an adipose cell, basically. We're trying to represent a cell. An adipose cell is an adipocyte. And here, this is a muscle cell. Okay, and the transporter in both these cases is Glute 4. Now, the importance of Glute 4 is that these are insulin dependent. Okay, so Glute 4 is insulin dependent. So what do we mean by that? We're going to explain now. So what happens is, these tissues, we said they are insulin dependent tissues. Okay, so there are also, let's draw this as a receptor for insulin. Okay, and let that be insulin. Okay, and 2 alpha units. And at the transmembrane domain, we say that it's got 2 beta units. Okay, so similarly here, there is an insulin receptor. And let's label this up. This is an insulin receptor. This is an insulin receptor. Okay, so through the blood, insulin comes into the adipose tissue. It combines with this receptor, the insulin receptor, it causes the attachment of insulin to the receptor, causes dimerization. Okay, it causes dimerization and phosphorylation. Okay, because there are tyrosine kinase domains here. Okay, so there are tyrosine kinases. Okay, anyway, there is dimerization, there is phosphorylation. The ultimate event is after that there is a signal going into the nucleus, and what the nucleus does is it makes lots of GLUT4. Okay, and also remember this GLUT4 which is present on the surface when there is it also goes into the cytoplasm. Okay, so when there is insulin available, then from the cytoplasm it goes into the membrane. So there is a constant cycling of the receptors. Okay, so when there is lots of insulin, then GLUT will be transported or translocated to the membrane. Okay, so there are normally lots of GLUT4 receptor present within the cytoplasm. On getting or attaching to insulin, those receptors go to the membrane. As well as new receptors, GLUT4 receptors are synthesized under the action of insulin. Okay, so all in all what happens is the number of GLUT4 receptors increase on the surface of the cell or on the cell membrane. So as a result, what happens? More and more glucose enters. Okay, so more and more glucose enters into the adipose cell. And again, this cell, what is the fate of the glucose that enters? It will undergo metabolism, or it will be converted to other components within the adipose tissue. Okay, and directly what it does, it increases the production or growth of it causes the growth of the adipose cells. Okay, so that is called lipotrophy. So the adipocytes undergo trophy, meaning they become big, and that is called hypertrophy. Okay, so they increase in size. Okay, now there is an applied aspect here. You remember we said that insulin is given by the subcutaneous route. Now under the skin there is lipids. Now if you give the insulin injection at the same site, what is going to happen is at that local area the lipids are going to increase in size. Okay. And when that happens on a continual basis, we call it lipodystrophy. Okay. And this kind of, if there is lipodystrophy, it changes the appearance of the body. Okay. So that is why the routes, you should not give insulin to this same place all the time. You need to rotate the location of administration. Okay, why are you doing that? To prevent lipodystrophy. Okay, all right, so that's the most important thing that happens with adipose tissue. But the important thing to remember is glucose is continuously getting into the cell. As a result, the blood glucose level is going to reduce. Okay, okay, in the muscle tissue, again the same thing happens. There is translocation of lots of GLUT4. Okay, so this is GLUT4, this is GLUT4, and that again is GLUT4. So what does GLUT4 do? It makes lots of insulin, and sugar enter, or sugar, in this case we are specifically talking about, it's a glucose transporter, so let's be specific. Let's call it by what it is, which is glucose. Okay, so glucose enters, okay, from all these places, glucose enters this cell. And here again what happens? Now this is a muscle tissue. Okay, so muscles, what do? They store glycogen. Okay, so excess of the glucose, some of them are used to produce ATP. Why is ATP required? Because muscles have to do work, and if they have to do work, they need to get the energy currency, which is ATP. So some of the glucose gets converted to ATP, and excess of it gets stored as glycogen. Okay. At the same time, there is decreased proteolysis, meaning synthesis of proteins is promoted. As a result, there is also hypertrophy of the muscles. Okay, so muscles also increase in size. So there is hypertrophy. Okay, there is storage of glycogen, and there is again production of ATP. Okay, so all these things happen in the muscle tissue. Okay, again, the muscle mass would increase, and that is again a anabolic process. The body weight would increase. Okay. Okay. There is one more effect that we need to remember about insulin. Okay. So insulin has got action on ions, specifically potassium ions. Okay. So what happens is insulin will push potassium ions, okay, into this cell. Okay, so potassium is an intracellular ion. And how does insulin do that? It stimulates the enzyme here. And what is that enzyme? That enzyme is sodium potassium ATPase. Okay, so by stimulating sodium potassium ATPase is stimulated. So insulin stimulates sodium potassium ATPase, which pushes potassium into the cell. So potassium, which was increased in the extracellular fluid, goes within this cell. And so what is the condition that there is increased potassium ion in the extracellular fluid? Yes, you're right. It is called hyperkalemia. Okay, so insulin has got effect like this. Therefore, it is used in the treatment of. Therefore, used in the treatment of hyperkalemia. Okay, because of this effect. But mind you, you're giving maybe a hyperkalemia, you're trying to treat hyperkalemia in a non-diabetic patient. Okay, so in that patient, if you give insulin, what is going to happen? It can cause hypoglycemia in the patient. Therefore, insulin should be administered along with dextrose. Okay, so usually, about 100 ml of 20% dextrose is used to administer insulin. And now you know why. Why should dextrose be given along with insulin to basically to prevent hypoglycemia. Okay? Okay. So now, what happens if you overdose a patient? Okay, overdose. Or the patient injects much more than necessary, overdose of insulin. What could happen? Okay, so this is a dose-dependent adverse effect. Okay, so what's going to happen is there's going to be hypoglycemia. So how can you correct hypoglycemia? Yes, by replacing dextrose. Okay. So replace dextrose. Okay, or give sugar. Okay, so the patient should be instructed, people who are on insulin should be instructed to carry candy. Okay, so dextrose, in case the patient has access to dextrose, you can give 20 grams of glucose, okay, which is about 3 tabs of glucose. Okay, 3 glucose tablets, or you could give maybe half a glass of juice, you know, like orange or apple juice. Okay. Or maybe half a glass of carbonated beverage. Okay. But mind you, it should not be sugar-free, okay? So containing sugar. Okay. If you use carbonated beverage, which is 0 calorie, like having aspartame instead of sugar, it might not work. Okay. Yeah. So this is how you could manage a person who is having hypoglycemia. Okay, if you have access to this. The other way you treat would be to give the counter-regulatory hormone. Counter-regulatory hormone. And which is the counter-regulatory hormone to insulin? You are absolutely right. It is glucagon. So if glucagon is administered, it can reverse the hypoglycemia caused by insulin. Okay. So, all in all, if we remember these aspects, we can easily discuss the pharmacology of insulin. Right? Okay, so let's put this piece of information back here in the graphic. We talked about the role of insulin. Okay, so insulin has got, in addition to the metabolic and anabolic effects, it has also got effect on potassium ion channels. Okay, yeah.